All right, today we are diving deep into the incredible world of genetics. We're going to tackle a massive question. How do we actually read the book of life? And we'll see it's really a tale of two very different stories. First, we'll jump into the wild, fast-paced world of bacteria, and then we'll get into the huge, complex epic that is our very own human genome. So this is really the core question for today. How on earth do you map out the genes of a tiny microbe that's swapping DNA like they're playing cards? And then how does that possibly compare to decoding our own genetic blueprint where, direct experiments are completely out of the question? The methods are just worlds apart, but as you're about to see, they are equally brilliant. Let's kick things off with bacteria. First stop, the microbial world. Now, you gotta forget everything you know about slow, generational inheritance. Bacteria are constantly swapping, stealing, and sharing genes. It's called horizontal gene transfer, and it's basically a genetic free-for-all. And it turns out, they've got three main ways of doing it. Okay, so the first method is called transformation. Just imagine a bacterium kind of hoovering up spare bits of DNA that are just floating around in its environment. Let's say a nearby bacterium dies, and its genetic code gets released. Well, another one can just absorb those fragments, stitch them into its own DNA, and boom, it's got new traits just like that. Next up is conjugation. You can think of this one as a direct bacterial handshake. One cell, the donor, physically connects to another one, the recipient, and passes a piece of its DNA right across. It's a very deliberate, one-way street for transferring genetic info. And finally, we have transduction. Now, this one involves a middleman that has no idea it's even involved. A virus. A specific type of virus, a bacteriophage, infects a bacterium. When it starts making copies of itself, it sometimes accidentally packages up a piece of the bacterium's DNA. So when that new virus goes and infects another cell, it acts like a tiny little taxi service, delivering the old bacterium's genes to a brand new host. So let's just quickly recap those three. We've got transformation, which is absorbing loose DNA from the environment. There's conjugation, a direct cell-to-cell transfer. And then transduction, which uses a virus as a delivery vehicle. Three totally different routes, but they all lead to the same result, a remixed bacterial genome. Okay, so with all of this gene-swapping chaos going on, you have to wonder, how on earth could scientists figure out where genes are actually located on a bacterial chromosome? I mean, it seems like it should be impossible, right? Well, the solution they came up with was just incredibly elegant. Yep, you heard me right. The key tools for one of the most important discoveries in microbial genetics were a stopwatch and, well, basically a kitchen blender. This brilliant experiment is called interrupted mating, and it let researchers map out the bacterial genome with stunning precision. Here's how this genius idea works. First, you mix donor and recipient bacteria together to get them to start that conjugation process. The second you do, you hit a stopwatch. Now, because the donor transfers its DNA in a nice, linear, orderly fashion, you can stop the process at different times by, throwing them in a blender for a second. By checking which genes made it over to the recipient cells after 1 minute, then 5 minutes, and 10, and so on, you can actually create a map of the gene order. The first gene to show up is closest to the start, and it just goes down the line from there. So simple, but absolutely brilliant. Now, let's completely switch gears, because mapping the human genome is a totally different ballgame. I mean, you can't exactly put people in a blender, right? This requires a whole different kind of science, less direct experimentation, and a lot more clever detective work. And this really gets to the heart of the matter. Since we can't set up controlled experiments, geneticists have to work with the data that nature gives us, our own families. So the question becomes, how can you possibly trace the inheritance of a gene for, let's say, a genetic disease through all the messy branches of a family tree? Well, the first tool in the Human Genetics Toolkit is pedigree analysis, and this is basically just creating a super detailed genetic family tree. By tracking a specific trait or a disease through multiple generations, scientists can start to see patterns. And those patterns help them figure out if a trait is dominant or recessive, or maybe linked to a sex chromosome. But just spotting a pattern in a family tree isn't quite enough. You need some hard statistical proof, and that's where something called the LOD score comes in. It's a way to answer the question, are these two things, like a disease and some other genetic marker, showing up together in this family just by random chance? Or is it because the genes for them are actual neighbors, physically linked on the same chromosome? The LOD score calculates the odds, giving scientists real statistical confidence. So what's the magic number they're looking for? In human genetics, a LOD score of 3.0 or higher is the gold standard. A score of 3 means the odds are literally 1,000 to 1 that the genes are linked, compared to it just being a coincidence. That's the kind of certainty scientists need to confidently say, yup, these two genes are definitely neighbors. Tracking patterns and calculating odds is one thing, but sometimes you just need to see the genes, or at least the chromosomes they're living on, with your own two eyes. This lets us go from abstract analysis to a concrete, visual diagnosis. And this brings us to the karyotype. You can think of it like a family portrait of your chromosomes. Scientists take a cell, get the chromosomes to show up, and then they digitally arrange them all neatly in order of size. This visual map lets them spot any big, large-scale issues at a single glance, like a missing chromosome, an extra one, or a huge chunk that's gone missing. And really, this is why all these techniques are so incredibly important. From pedigree analysis to karyotyping, these are the critical tools we use for diagnosing a whole range of genetic disorders. They let us find the genetic basis for conditions like cystic fibrosis and Huntington's, and that gives families vital information about their health. So let's just bring it all together. On one side, you've got microbial genetics. You're dealing with crazy fast gene transfer in bacteria, which you can study with direct, hands-on methods like interrupted mating. And on the other side, you've got human genetics. Here, you can't experiment directly, so you have to rely on the clever detective work of pedigree charts and some really powerful statistical analysis. So from a simple blender and a stopwatch, all the way to complex statistical models, we've built this incredible toolkit for reading the book of life at every single scale. This knowledge has completely changed medicine and how we understand ourselves. The final question, then, isn't just about reading, it's about writing. What will the next chapter hold? Thanks for joining me.